So before we start on Chapter 18, do people have any questions left from Chapter 17 and the general notion of DNA synthesis and of DNA packaging and the C-value paradox? Any general questions? Questions about the course overall at the moment? Okay. All right. Let's proceed then with Chapter 18. Okay, let's try that. There we go. And so in Chapter 17, we spent a lot of time thinking about making copies of DNA and the challenge that presents, and packaging DNA, sort of in between time, so to speak. But the fraction of time that is actually spent being replicated is very small for a DNA molecule. In a population of rapidly dividing cells, maybe 10 or 15% of the time. In a cell like your muscle cells or your neurons, most of those are not making DNA at all. So most of the time, a DNA molecule is not confronting the issue of being replicated or not. It's confronting the question of staying intact. And so what we'll be exploring in Chapter 18 is the other side of DNA metabolism, which is DNA repair, and how that couples to another very important medical question, which is cancer. Cancer is one of these words where you can use one word and mean any of many different things. Cancers can vary from a benign tumor that is, slowly growing over 20 years and you die of something else first, to galloping pancreatic cancer that kills you in a matter of weeks. But whichever form of cancer, whichever tissue and however malignant, in general, cancer is associated with some form of problems in DNA. It's at its ground, it is a kind of mistakes in the DNA that have consequences. And those come in any of several places. One is in DNA replication, when mistakes are made, mistakes in repairing it, and physical damage. And all those things can have consequences to the integrity of the DNA, and therefore specific consequences to the integrity of the ongoing developmental program or the ongoing maintenance program of that particular cell type. This is showing you one particular cancer cell that has almost detached from the cell's surface. So instead of being a nicely behaved cell that's spread flat and intimately connected to the surface that it's supposed to be growing on, it's lost a lot of that element of connection, and it's getting ready to be completely independent of restraint of that kind. DNA damage can happen from a number of sources. We'll be talking about some of the specifics in a bit. And so all organisms have some mechanisms of DNA repair, some way of coping with damage to the DNA. And to give you a sense of just how powerful that DNA repair can be, I want to share with you this microorganism called Deinococcus radiodurans, which means monster bug, monster coccus, who's tough to radiation. And Deinococcus radiodurans is the most resistant to radioactivity of any organism known. So if we take one particular unit from talking about radiation, there are several. 10 grays of radiation will kill a human being. E. coli is a bit more resistant. It takes 50 grays, 5 times as much radioactivity to kill E. coli. 100 times that, 5,000 grays, will not even affect radiodurans. And this resistance to damage is exemplified under a number of different contexts. Radiation damage is how it was first discovered. It was in the early days of trying to explore using radiation to purify or to sterilize foods. And they would open the cans, and it'd be full of this gray stuff that clearly had survived doses of radiation that should have killed them. But the resilience of Deinococcus is beyond just the damage due to radiation, because it can resist acid, it can resist vacuum, it can resist cold. Its repair resilience works across the spectrum of activities. These are micrographs of Deinococcus. This is a light micrograph showing you that it usually grows in this 4-cell cluster that's characteristic for coccus. You can see in this one what's called a transmission electron micrograph, and you can see the density and the organization inside these cells. These aren't just kind of random bundles of stuff. We're going to see that this is a very well organized genome. So I mentioned that it was discovered by accident because when they sterilized food, this thing didn't get killed. It's being explored as a tool for cleaning up radioactive waste. Bioremediation of various kinds of environmental damage is being explored for a number of different environmental insults, but radioactivity obviously presents a specific challenge, and Deinococcus is pre-adapted to something like that. And part of what makes Deinococcus so powerful is that it has multiple copies of its genome, its DNA, that are bundled together to maintain them in a fairly close relationship. But the other part of it is that it has extraordinarily fast DNA repair mechanisms. And these two electron micrographs, these are now looking at just the DNA molecules. And this top panel here is the DNA molecules immediately after that irradiation event. And it looks like polka dots or, Swiss, dotted Swiss or something. And here is the reannealed, reconstructed, regenerated DNA molecule 12 to 16 hours later. So an extraordinary capacity to repair damage that would, by all intuitions, say could not conceivably be repaired. They're very good at it. We're not nearly as good at it. And one of the consequences is that we, speaking of humans generically, can undergo that change of phenotype, that change in differentiation that's called cancer. Cancer is really many different kinds of things. So a cancer of the liver will present differently than a cancer of the muscle, will present differently than a skin cancer. But the common feature that they all share is that they have uncontrolled growth of cells. Now, that doesn't mean fast growth. Some cancers grow quite slowly, but their growth is not controlled in the way that other cells of their type are controlled, and so it gets out of the bounds of the appropriate cell type. Another characteristic of cancer cells is they fail to die. Normal cells have, in amongst their various programs to do things, a program to die called apoptosis, and this is triggered by certain specific environmental cues that, in the estimation of the body, are good ones to get rid of that cell. And it triggers a program of self-destruction, and the cell is gone. Cancer cells have that program turned off. They're deficient in it, and so when the body tries to get them to die, they don't. These are always associated with alterations in the DNA sequence. To give you a little bit of a sense of dimensions, there were almost 16,000 new cases of cancer in Iowa, just Iowa, in 2006. This is a photomicrograph of a liver. You can see here the individual tumors in this liver. I don't know from this one whether these are metastases from somewhere else or whether they're primary to the liver, but you can tell by looking at it that this is not a healthy, happy liver. So what kinds of things can cause cancer? In terms of sources, you can roughly divide it into heredity and environment. Some of the environmental cues are things like smoking. No surprise there. Although the tobacco companies fought this piece of scientific reality for a long time, it's pretty unequivocal now. Sunlight can contribute to skin cancers. There are some viruses that can cause cancer. So, for instance, human papillomavirus will cause cervical cancer. A herpes virus, I believe it is, causes Burkitt's lymphoma. A small number, not nearly as many as the virologists were hoping for back in the 70s, but there are some cancers caused by viruses. Asbestos. Asbestos is actually not itself a DNA damaging agent so much as a selective agent. That is, it's a long, skinny thing to grow on, and only cells that have had their DNA damaged will grow on it. So it provides for a selective filter for cells that have been damaged. Nitrosamines, these are things that can damage the DNA chemically. You may run into them in those delicious things called hot dogs, some other things that are preserved with nitrites. So there are a variety of different things, almost all of which share the feature of being DNA damaging agents. We'll see in a little bit the kind of damage that UV light, for instance, does. Smoking does several kinds, and so on. That's the environment side. The hereditary side, there are a number of genetic phenotypes, human gene changes that predispose people to cancer. They are not per se causative of cancer, but they are in genes that are needed in order to repair DNA damage. And so people who have those mutations aren't very good at repairing the damage that's going on all the time, and so they have a higher frequency of cancer for that reason. So that's in terms of sources. What kinds of things go wrong in cancers? And we roughly can divide those into genes that are too active, that are more active than they would be in a normal cell, and cells that are less active or inactive compared to a normal cell. So damage can lead to either kind of change, and we'll look at some of the specifics on that. When we talk about the genetic basis for cancer, now we're talking about mutations that you either were born with or that you acquired environmentally. We divide them into two largish groups. When we talk about proto-oncogenes, these are genes that are present in normal cells, and in normal cells they're going to control things like cell growth, cell differentiation, and they're called proto-oncogenes because if that gene is upregulated so it's more active than it should be, then it's going to lead to a cancer, and that's where it gets its name from. Oncogene means cancer gene. So it's named for what happens under extraordinary circumstances, but it has a normal, healthy role in normal cells. Those are the ones that cause cancer by being too active. The other big group of cells, or genes rather, are tumor suppressor genes. These are genes that would normally be active in normal cells, but if they are lost, the cells then progress into some state of cancer, carcinogenesis being cancer formation. And this is just a diagrammatic representation of that. In a normal cell, there will be a particular balance between the proto-oncogenes and the tumor suppressor genes, such that there's more of a signal from the tumor suppressors than the proto-oncogenes, that overall cell growth is maintained at the right level, cell differentiation is sustained at the appropriate level, and so on. Transformation is the generic term we use for turning a normal cell into a cancer cell. And at the end of transformation, one of two things is going to be true. Either there will be proto-oncogenes that have been activated and are now oncogenes, or tumor suppressor genes have been turned off and there's not enough activity from them. So one way or the other, you're going to have shifted the balance towards the side of favoring cancer as opposed to favoring the normal state. These are some terms you're likely to hear at various times. Carcinogenesis, okay, carcin, that stem is cancer-related. Comes from the word for crab, that a cancer growing in a tissue look, at least to the ancient Greeks, like the claws of a crab growing in and grabbing the normal tissue. Genesis, of course, the becoming of something. So that's the development of cancer. Transformation, as I said, is what we call the conversion of normal cells to cancer cells. Then a tumor is simply a mass of cancer cells. This is sort of a schematic of some of the things that are going on. There is always a first step, the first step down the lily path. Initiation has to happen through some kind of an event, typically a carcinogen, that creates a permanent genetic change in that cell. Promotion is a blanket term covering a variety of things that can encourage cell multiplication. They can be chemical agents. They can be other mutations. Typically, promoters are chemical agents that won't by themselves cause cancer unless other mutations have happened. With further genetic changes, you now have a transformed cell, a malignant cell, that is independent of the normal constraints of growth, but that's not the end of it. Cells go on changing once they become transformed, and in fact, cancer is an extremely diverse tissue, speaking genetically. There's a wide variety of genotypes in that tissue because cancer cells have a higher mutation rate than normal cells. And so they, in a malignant tumor, there will be many different micro colonies of different cancer cells. So some of the kinds of DNA damage that can happen, there can be a base substitution, so that during the replication process, maybe the wrong base was put on and it wasn't catched by the repair. There can be deletions of stretches so that you're actually missing a little bit of a gene. You may have modified it incorrectly. Remember, we were talking about methyl G and so on. If you incorrectly modify a gene, it can be as serious as changing it. There are some heritable conditions that contribute to this process. In general, there are going to be more than one pathway affected. It's not enough to do just one gene mutation. This is sort of a very schematic idea that you're born, you have beautiful, lovely, pristine cells for the most part. A cell gets one mutation, gets a second mutation in a different gene, gets a third mutation in yet another gene, and a fourth. There's some number, we don't know what that exact number is, between 5 and 10, that is necessary to happen before you actually have a transformed cell. It's important to remember that which gene mutates first does not matter. It's the total number of changes, that you've hit enough different regulatory pathways, that you've knocked out this regulatory pathway and this one and that one. So there's multiple genetic changes in any cancer tumor. And some of the things that are typically found to be altered in cancer cell are things like the regulation of cell division. Mitogens is a generic term for growth factors that cause cell division, and they are used to signal to a cell grow. Typically in a cancer, the growth signaling pathways are stuck in the on position. They're in a normal cell, there's an on state and an off state, and you'll be on for a bit and then off most of the time. In cancers, the signaling pathway, through a mutation, is now stuck in the on signal. A second aspect of cell regulation that's important is apoptosis, this programmed death that we talked about. And any given particular tissue type has certain signals that are, for that tissue, the appropriate signal that die. It's time to die. It's true. And part of being a cancer cell is they no longer hear those signals. They are resistant to those signals, and therefore they go on growing at a time when they should be dying. A third factor that is an aspect of maintaining normal regulation is the maintenance of telomeres. And I think you may remember that I said that at some point during normal aging and so forth, the telomerase, the enzyme that makes this stuff, the activity starts to fall. Cancer cells in general will have an activated telomerase that is as active as a newborn infant or more active. And so they have telomeres that can stand up to many rounds of division without going away. Another process that is changed in cancer cells is the normal relationship to the geometry of growth. Normal cells have two things that are aspects of how they sense their environment and respond to it appropriately. It's called contact inhibition and anchorage dependence. So contact inhibition means when the two normal cells are growing and growing and they find another cell and touch it, that contact in a healthy cell will typically inhibit further growth. The second measure of that is normal cells need something to hold on to while they're walking and dividing and so forth. That's called anchorage dependence. If you don't need something to hold on to, now you're anchorage independent, you could grow whether you're up in the solution or not. And these are two pictures that show aspects of this. In this panel of normal cells, you can see that for the most part, the cells are not on top of each other. They've touched and stopped, that's contact inhibition, and that they're spread pretty well on the surface. They're dependent on that anchorage for proper cellular metabolism. These cancer cells, by contrast, are growing stacked several deep. They're not well attached, and in fact, some of these look like they're ready to float off and just grow in the medium. One of the most famous cancer lines, called HeLa cells, has grown in, some strains of HeLa cell have grown in suspension for 40 years without ever being attached. And this is important for a couple of reasons, because cancer cells that are not contact inhibited can crowd. They can make it really continue to grow even when the area is kind of crowded in, and they are pre-adapted for metastasis. That is, if this little cell here that's about to float away gets into the bloodstream, the fact that it's anchorage independent means it'll still be happy floating around the body until it finds a new niche where it can sit down and start and make a new tumor. So these are also important properties of normal cells that are lost during this sequence of mutations. And again, the sequence in which these losses happen is not important. What is important is that all of these processes usually are dinged in a cancer line. Another dimension of cancers that's becoming more and more appreciated is that they are capable of doing what's called angiogenesis. Genesis, making something. Angio is blood vessels. And so if a small tumor forms and it's just there, it's hit, had these other pathways hit, but it doesn't yet have angiogenesis, it'll make a little nubbin of a tumor, but it'll starve. It won't be able to get much bigger because it won't have a supply of blood to it. And it was the insight of Judah Folkman and others like him that at some point a cell in that colony develops the ability to send out the growth signals to attract blood vessels, and once it can do that, it can attract its own blood supply, and now it becomes a parasite on the body. The blood supply is coming to the tumor, it's feeding the cells, and now they can keep growing because the blood supply will bring in nutrients and take away their waste products. Until angiogenesis is sustained, you'll find that the center of a small tumor is necrotic. It's dying from its own waste products and for lack of food. Once a tumor has blood supply, it can grow like a miniature organ. It has no purpose to the body, but it can develop and grow. So that you can see, I talked about 5 different characteristics there, so you can see why it takes at least 5 mutations. Whether it's exactly 5 or 7 or 10 will be an area of continued research, but you can see that there's different kinds of things that have to be lost or gained before a tumor cell can actually be a fully independent, vicious, nasty cancer cell. So even though there's great diversity among cancers, you have many different tissue types, you have many different growth rates, there are some features that are common to cancers, and those were the areas in which there was the first effort to target cancer with anti-cancer agents. Obviously, if we define a cancer as uncontrolled cell growth, things that suppress cell proliferation should suppress cancer cells. These might be things that can target some of the growth signals, break past the damaged signaling pathway and send in a signal. This is one particular anti-cancer agent that is kind of a cute one, because this was one of the first molecular design drugs where they looked at a protein and designed something to fit into it. And it targets something called BCR-ABL, which is a tyrosine kinase. It's associated with a kind of leukemia called chronic myelogenous leukemia. So these are people who are casting off white blood cells at higher levels than they should be on a chronic basis, as opposed to acute myelogenous leukemia, which is very bad stuff. And Gleevec can bind to this mutated kinase. So BCR-ABL is a translocation that puts together parts of 2 genes. And Gleevec is specific for that hybrid molecule, and so it's relatively benign to the rest of the body. Unfortunately, like almost all drugs, eventually cells become resistant to it, but it's a beginning. There are a variety of ways that you can inhibit cell division. We talked back in the cytoskeleton chapter about how microtubules are necessary for cell division, for mitosis, and a number of the anti-cancer drugs are actually antimicrotubule agents. One difficulty with that is that it's not a very specific thing, and the most rapidly dividing cells in the body will be hit by it as well. This is one of the reasons why anti-cancer treatments so often hit the hair, the gut, and so on, that the hair and the lining of the intestines are both tissues that have a fairly rapid growth rate by comparison to the rest of our body. So anti-cell division agents tend to hit them in addition to the cancer as well. We can use radiation. We are not Deinococcus radiodurans. We don't survive huge amounts of radiation. And the newest machinery for this has gotten extremely ingenious, where they'll rotate the radiation head around the target like this. And so any one path along the way doesn't get that much radiation, but the point that's common to all of them is the tumor. So the tumor may get 100 or 1,000 times as much radiation as the rest of the body does. And radiation is even less specific than inhibiting cell division. Radiation is just slamming on all the molecules of the cell with sledgehammers. I want to put in a pitch for healthy living again. You've heard me do this back when we were talking about diabetes. It is estimated that half of cancer deaths could be prevented. It's far cheaper to prevent a cancer death than to treat a cancer. So any cancer death that is prevented, you've got all the extra healthiness of the life in the meantime, and you haven't spent that money. I hope you all know that if you're smoking, you should stop. Or chewing tobacco. A major cause of cancer. Moderate sunlight. You don't have to be a caveman. High exposure to the sun is associated with melanoma. A little bit of sunlight actually is good for you. It makes vitamin D, and vitamin D is an anti-cancer agent. Healthy eating. There's all kinds of peculiar compounds in vegetables that turn out to be anti-cancer agents. And so far, we really haven't gotten them into pills in any very good way. And since they're also good for you for other reasons, developing a taste for fruits and vegetables has many payoffs. Reducing chemical exposure. California is an interesting state because it's a state law that anything that is at all considered a carcinogen has to be posted on the building. And so you wander around in California and half the buildings have, warning, there are things in this building that are being used that could cause cancer to you. Of course, you don't know what they are. So. You need to be a little bit prudent about it and pay attention, but there are chemicals that can increase that. There are some microorganisms that can cause cancer. So, for example, one of the forms of hepatitis can cause liver cancer. The organism that causes ulcers also causes stomach cancer. So the kinds of public hygiene things that reduce those agents can also reduce cancer as well. Sermon over. I've mentioned several times now that cancers are very diverse, and we're getting more tools for trying to get a handle on that diversity. And one of the newer ones is something called gene profiling. And this is an intriguing sort of tool for going about it with the ability to make probes of known sequence and so forth. We can now identify levels of expression for hundreds, thousands of genes at a time with a single gene chip. And this allows us to characterize different kinds of cancers as having different profiles of expression for different genes. Using this needs to be a little bit subtle. In the early days, people would just take a chunk of tumor and put it in, and since that chunk of tumor had maybe 10 or 20 colonies of tumor cells in it, the results were not as informative as they might have been. With careful dissection, so you know that you're sampling it discretely, it's probably a little bit better. The hope is that this was going to give us improved diagnosis, and we are beginning already to notice that there are some patterns of expression that have worse prognosis than others, which is, one help. The further help is as we get to understand better the specific patterns of these subsets of tumors and so forth, we'll start to have better treatments. And I think that's still a little early, but it's a reasonable hope to have. So where does this come from? Where do these gene alterations come from that give rise to cancer? I hope I've motivated you that DNA repair is important. Well, there are a couple of ones that can happen. There can be actual physical damage to the DNA, and there can be mutations in the DNA that arise through the process of replication. And we're going to look and see how the cell actually can manage those damage. There are specific repair mechanisms, and there are enzymes that can deal with damage. This is a very sort of generic representation of the idea that you have some form of damage here that's recognized, taken out, and re-annealed. That's sort of the generic strategy that's going to be there. So some of the environmental sources of DNA that I mentioned earlier today include chemical mutagens, things that chemically react with bases. Ionizing radiation are the kinds of radiation on the electromagnetic spectrum that when they penetrate a tissue, if they interact with a molecule, they can generate ions at that spot, and those ions then in turn can cause damage. UV light has a very specific kind of damage it causes. It's like twanging a harp. It causes thymine dimers, two thymine bases that are side by side or one on top of each other in the DNA, to form cross-links. And this is called a thymine dimer here. And you can see now that they're, first of all, no longer independent of each other, but they also cause a kind of a kink in the backbone at that spot. And so this would obviously not be a good thing to try to transcribe or replicate through. Some of the other compounds that can cause mutations. Here's benzopyrene, very aromatic compound. We find benzopyrene in cigarette smoke, in car exhaust, and it can become covalently attached to DNA bases and interfere with things. Here's aflatoxin. This is a byproduct of molds. It's one of the reasons why a good Food and Drug Administration is good, because you want healthy foods. Again, can make base addicts. Just the fact of living can be bad for DNA. So the fact that we have an aerobic metabolism means that there is some number of oxygen radicals that are formed in the course of metabolism. We have a good control over it, but not a perfect control of it. And an oxygen radical like this with an unpaired electron there can become covalently attached on a guanine, and this forms a modified base called 8-oxoguanine. And this is one of the commoner forms of chemically modified bases as a consequence of environmental assaults. And the problem with 8-oxoguanine is that it's modified the potential in here for hydrogen bonding in such a way that it can be base paired with either C or with A. And so it can induce a change in the DNA sequence if you go through a round of replication and put in an A instead of a C, and the next round you're going to put a T opposite that A, you've changed that gene. And that creates what's called a point mutation at that one point, that particular base. Other kinds of damage just happen just because we're at ambient temperatures. The bond between the ribose, the deoxyribose ring, and the base can hydrolyze. You see here this little alcohol there. Where that alcohol is, it should have been a base. That leaves you with what's called an A-basic site. Obviously, it doesn't know what to base pair with, right? Another spontaneous damage that can occur is you can have a cytosine that spontaneously loses the amine group oxidatively. And now what you've done is you've created a uracil in your DNA. And the uracil then would look like a thymine, and that would, again, switch which base pair would be used for replication or transcription. So these are multiple sources that can cause DNA damage. And if those are not repaired, then those different things can alter what's going to be made on the encoded protein. So just to summarize, you can have a point mutation where you change which base is at that site. You can have insertions where you've added a little bit of DNA. You can have deletions where you've taken a little bit out. If the damage is sufficiently serious, the cell can detect that, and it will stop replication, and it will stop transcription. And sufficiently serious DNA damage is one of the things that can trigger apoptosis. So in a cell that still has functioning apoptosis, sufficient damage will likely lead to cell death at that point. If the cell does not still have functioning apoptosis, that kind of damage may lead to transformation. It may have wiped out some of the important things for the rest of the thing to go on. And so that's why we have DNA repair mechanisms to cope with all that. So let me just check on something here. Okay. There are a number of repair enzymes that are worth paying a little attention to. Some of them are very simple. Here's your thymine dimer again. This was formed by UV light going through two thymines side by side. Bacteria have a DNA photolyase, which is kind of insidious because it uses UV light to activate that bond and then repair it. So it's kind of like you got your cure where you got the disease to begin with. Another repair enzyme is one in mammals where we can correct methylguanine and transfer that methyl group and put it onto an amino acid to restore the guanine to its correct state. But there are other things that require more complicated a sequence of events before you can repair the damage. Some of the ones we're going to be looking at in a little more detail are mismatch repair, base excision repair, and nucleotide excision repair. And again, this is sort of your generic one-size-fits-all schematic of what's going to go on. So the mismatch repair system is called MUT-S, and MUT-S is going to look at newly made DNA. It's in the process of just going around and scanning DNA that's been recently replicated just to sort of follow up after the replication machinery and just keep a look at everything that's been recently made. And what it can do is it will recognize and bind at any place where it finds mismatched bases. So if that highly accurate DNA polymerase, that's really very good, makes a mistake, that leaves a mismatch, then MUT-S will identify that and stay bound there. The next step is a nuclease that's going to cleave the DNA, and it's going to target that cleavage to the strand with the mismatch, which you might ask, how do you know is the wrong side just by looking at it? And this is one of the benefits of methylation. You remember we talked about being able to modify DNA by methylating some number of the bases so that a newly replicated molecule of DNA has one strand fully methylated and one that has not yet gotten methylated. So the new strand is going to be the strand that's not methylated. And that's how the enzyme can recognize that's the one to target and not the other one. The helix is unwound at that site, and the base is removed and then repaired. And you can see how the MUT-S is kind of surrounding the DNA, looking at the goodness of fit. If you think about the beautiful, smooth, double helical character of DNA, you can imagine how a mismatch is going to either make it bulge out or smush in. So all it has to kind of do is wander along that looking for something that's distorted. Base excision repair is another mechanism that's available to the cell. It can attack a number of different kinds of damaged DNA, and it can recognize sites of damage. It will go in and generate an abasic site. That is, it will deliberately cut off the base. And that leaves then an abasic site, which has nothing across the strand from its other base. An endonuclease can clip the DNA backbone at that spot. And then DNA polymerase and DNA ligase can close that rest of that in to seal that gap. Base excision repair can target a variety of damaged bases. So let's take a little bit of a look at 8-oxoguanine DNA glycosylase. So you may remember I just mentioned that one quite common form of damage to DNA is just a spontaneous side effect of the fact that we're aerobic, that oxygen radicals can attack guanine and make 8-oxoguanine. And if we look here at the two configurations of what would be found on the DNA, this is the 8-oxoguanine base that's needs to be repaired, and we're going to have to somehow or other get it out of the way so that we can attack it. The protein is capable of identifying that as an 8-oxoguanine and flipping it out of the double helix. So you can see how it's now no longer in the context that it was before, but it's still being stabilized by here an arginine and a cysteine that are bonding to it. So in this flipped out configuration, it's sort of lying out there waiting to get cut and available for that excision. So another form of base excision repair is a uracil DNA glycosylase. You remember we said that the oxidative deamination could convert a C to a U, and you do not want uracils in your DNA. So if you find a uracil, you know it's wrong. That those uracils might have gotten there through the cytosine deamination I mentioned. They might have gotten there as just a mistake. Either way, you don't want them out there. And the glycosylase in this case is going to stabilize the transition state. The phosphates of the DNA are going to interact with the ribose and allow that uracil to be cut away. Okay. I think we'll call that it for the moment, and I'll see you on Wednesday.